Have you ever wondered if humans could slow down the age? If all our advanced technology, developed scientific understanding, or our nutritious diet could slow down, or even reverse the seemingly simple process of aging? Running marathons and intensely exercising every day give you a better chance to live longer. Well, this can simply be answered by science and its analysis of a teensy little thing called telomeres. Telomeres can be described as the cap at the end of each strand of DNA that essentially protects our chromosomes, like the plastic tips at the end of shoelaces. Without the coating, shoelaces become frayed until they can no longer do their job. Without telomeres, DNA strands become damaged and do not divide properly, causing ends to be cut off. And thus our cells aren't able to do their job correctly, as DNA makes up all the cells of the body containing the genetic material that makes us who we are. Telomeres are vital for our health. Aging represents the accumulation of changes in a human being over time, encompassing physical, psychological, and social changes related to growth. Since our cells divide constantly throughout life and DNA strands become slightly shorter each time a chromosome replicates itself, it means the telomeres that help prevent genes from being lost in this process become consequently shortened. That's where an enzyme called telomerase comes in. Telomerase is found in certain cells and helps to prevent the wear and tear within the chromosome, including the shortening of telomeres by adding additional telomere sequence to the end of chromosomes. However, most of the cell types in the body won't have telomerase, and most telomeres will continue to shorten over time. Scientists now have more of an understanding and refined knowledge of telomeres compared to that in the past, and continue to find new evidence of the roles that telomeres play in the aging process on the cellular level. However, recently the idea that high-intensity exercise could preserve telomere length and slow down aging by almost a decade has risen, with scientists conducting a variety of investigations to come to.
So what exactly is high-intensity exercise? Well, it is defined as physical activity performed with a large amount of effort, essentially resulting in a higher heart rate and rapid breathing. This can include weightlifting, marathon running, and even jogging. While everyone knows exercise has short and long-term benefits for our health, scientists have recently discovered that it has an effect, while minimal, on telomeres and their length. Investigations done by multiple articles, such as our primary and secondary articles, found that telomere length was better preserved in older endurance-trained athletes than the same age group with a medium level of activity. But surprisingly, there was no present association with exercise and telomere length in younger individuals. Telomeres shorten with age, and this progressive shortening leads to senescence, apoptosis, or oncogenic transformation of somatic cells, affecting the health and lifespan of an individual. Regular high-intensity exercise is thought to reverse the effects or even slow down this process of shortening telomere length, and systematically slows down aging by almost a decade. There is some evidence that has shown that this type of exercise can reduce chronic inflammation, which is long-lasting swelling, as well as oxidative stress, which is an imbalance between the reactive oxygen species and the ability to readily detoxify the reactive intermediates and repair the resulting damage, especially in older, obese individuals, and may thus reduce telomere shortening observed with aging.
This research, however, is inconsistent and raises many questions within the scientific community. This is mainly due to the variety of results received by multiple experiments and their conclusions, with some finding that telomeres can be greatly shortened with exercise and others finding no evidence at all. This raises the question, are there other factors affecting telomere length? As previously mentioned, there is a distinct association with telomere length and age, with telomere length in older individuals being affected by this type of exercise, while younger individuals were not. However, there may be a substantial number of factors, both modifiable and non-modifiable, that could significantly affect an individual's age and telomere length. Telomere length is affected by a combination of factors, including age, genetics, environment, social and economic status, body weight, smoking, and many more. With this in mind, scientists are currently unable to provide accurate and correct evidence to conclude that an individual's age can be slowed down through high-intensity exercise alone. Despite the ongoing hunt to discover the truth, there are also many limitations which make us unable to determine the truth behind telomere length and exercise. For one, many primary articles investigated using different methods to select samples, determine type and duration of exercise, and to test the length of telomeres. However, many of the primary data experiments are between athletes and inactive people. This is significant, as athletes usually live a distinct lifestyle with healthy eating and sleeping patterns in order to maximize their performance and health, something that many others would find impossible to achieve. Furthermore, athletes may have inherent traits which make them elites of their field. This discrepancy creates significant variables which may have a huge impact on an individual's telomere length.
In this experiment done by Awama et al. 1998, the researchers investigated telomerase activity and telomere length of peripheral blood mononuclear cells obtained from 124 healthy individuals aged between 4 and 95 years of age. Telomerase activity is assessed semi-quantitatively by a fluorescent telomeric repeat amplification protocol. This is basically a tool that was recently developed to detect telomerase activity in cellular protein extracts, where telomerase synthesizes a specific repeating nucleotide sequence onto the ends of telomeres, identifying telomeric repeats. Telomere length is measured by terminal restriction fragments, TRF for short, in DNA by a Southern blot analysis. This process is the oldest approach of analyzing telomere lengths and remains the gold standard even in current studies. This technique relies on the fact that repeated telomeric units do not contain target sites for restriction enzymes. So what exactly do these results mean? Well, it essentially proved that the decline in telomere length differed as individuals got older, with the telomere reduction rate being substantially greater than that of the elderly individuals. As for the experiment involving telomerase activity, the results show that telomerase activity in normal individuals aged 40 years or under gradually declined with increased age until after 40 years old. It is seen that after 40 years, there was a very low level or undetectable level of telomerase activity, essentially demonstrating how telomerase activity declines with aging. This investigation, however, contains many limitations. One of the major limitations involves the technique used in assessing telomere length, as it could not accurately measure the exact length. Instead, it provides a rough estimate, particularly in measuring that of the elderly individuals, leading to inconsistent and inaccurate data. Furthermore, an explanation for the reduction of telomere length is not provided in this study, leading to an uncertain conclusion to whether aging is affected by intense physical activity.
Our topic and our presentation greatly aligns with certain themes in this course. As previously mentioned, this topic is surrounded by many uncertainties and inconsistent information, providing evidence on how to think like a scientist. Over the course of our research on high-intensity exercise and its effect on aging, it has been apparent that scientists communicate by spreading information through journals, articles, and scientific meetings, discussing the concept at hand. Through this, biological scientists and researchers have concluded that there is not enough data or evidence to come to an overall conclusion that high-intensity exercise slows down aging by a decade. It is through the updated investigations and continued communication that allows us to understand that this is essentially a never-ending exploration, and communication of investigations will always be used to present findings to other scientists as well as the broader audience. We have also ensured all information included in this presentation has been found in reliable articles and real scientific journals to ensure all research is true and plausible. This presentation presents cell structure in depth, focusing strongly on telomeres and their effect on the body. We have delved into the details of cells and the cells that cause a delay in the function of growth. Throughout this process, we have also understood that cells are not affected by one factor only, and are actually influenced structurally and behaviorally by several factors, such as lifestyle behaviors and non-modifiable factors. Past lectures and practical lab lessons on cells presented insight into the different types of living cells, as well as the similarities and differences amongst them, that cause them to function differently. In this case, Y telomeres within cells are affected by high-intensity exercise, so much so that it may systematically slow down the shortening process which affects the aging process of an individual.
Overall, we are able to say that there is a connection between telomere length and high-intensity exercise, even if it is difficult to distinguish. This is presented through past research, showing those who regularly exercised at high intensity have longer telomeres than those of their age who didn't. However, the maximum that telomeres can be lengthened and how long they stay lengthened is yet to be answered. So it is safe to say that exercising is beneficial nonetheless, and could potentially slow down the process of aging. But watch out, this is not to say you should jump up and immediately start exercising. Because if you are lazy, like us, sudden exercise might actually cause injury rather than burn off your age count. Nevertheless, now there is no excuse for not exercising more often.